in and get a seat. We have never had this room quite this full before for one of our programs, so this is really exciting. And the median age is not 60 years old, so that's just fabulous. So welcome, welcome, everybody who is a member, isn't a member. So far, we don't have any on Zoom, but we'll be getting some on Zoom. My name is Mayo Taylor. I'm president of the Blue Ridge Naturalist Network. And for the many of you who aren't involved with our group, we are an outgrowth of the Blue Ridge Naturalist Certificate Program at the North Carolina Arboretum, which is a fairly rigorous set of classes that you take to be certified out there. So should you want to get more involved, I'll give you a couple of ways to do it. But I wanted to get us going fast because I have a feeling that some people want to be someplace else at 9 o'clock for some other event that's going on. So I hope we won't have to belabor this at every meeting, but if you have not set up your parking pass, your free daily parking pass, you probably ought to do it. I have no idea what the repercussions would be if you didn't, but we've got computers over here on the side, or you can do it on your phone if you Google around to find UNCA parking pass. It's a new thing. And there's a QR code. All right, a few announcements to do at the front. On September 26th, we will have our 11th annual meeting, and it's going to be out at the Arboretum. It's going to start with some guided walks in the Arboretum. One of them will be a mushroom walk by Charlotte Kaplan. And then Randy Richardson and David Tifatiller are also lined up to do it. The annual meeting is normally only open to our own members, but we are going to have some extra people coming in this year, hopefully, which will be the currently active students in the Blue Ridge certificate program. So we're hoping that in that way, we're going to identify the new people who have gotten into that program and are looking for things to do with their newfound skills and knowledge. The annual meeting is usually open, it is open to members. If any of you are not a member, but you would like to be at that annual meeting, we have a deal for you, which is that as of September 1st, if you go ahead and join up as a 2025 member, you get this fall, and then you get all the way through next year, and then you'll get an invitation to come to the annual meeting. So we've got membership forms over there. If you're not ready to get quite in that far, we have a list over there to get on our newsletter, which goes out to anybody who wants to get the newsletter. And then you hear about all our activities, and then you get really excited about joining up and being at our 12th annual meeting. So it all works together. We have one more public program this year. It will be here on the second Tuesday of October, which is the 8th. And it's Pam Torlina, who is the communications director for Conserving Carolina. And if you don't know, Conserving Carolina is just a hyperactive conservancy that works primarily in the counties south of us, but also into South Carolina. And they do a lot of unusual things for conservancies. They do a lot of public access to their properties and a lot of public programming, educational programming. And she will talk generally about all the many threads that they have going, but she's particularly going to look at, they have an incredibly ambitious overall plan for the Hickory Nut Gorge for special lands to protect and up to 70 miles of trails to connect them. So a lot going on there. So that's October 8th with Pam Torlina. We have, in addition to the guided walks at the annual meeting, we have at least one more outing for the year, and that will be to purchase knob. I know some of our members will be glad to hear that, and the people responsible for that just walked in the door, so that's excellent. Marilyn and Lou will be leading that one. The date on that is October 1st. That's a Tuesday, and the rain date will be October 3rd, and of course you'll get the usual notifications on that. All right. I'm going to introduce our speaker. Before I do, let me say that hopefully we are going to have some Zoom people. And so it's much easier for managing that if you hold questions to the end. If any questions get asked during it, you can just repeat them. And then for those of you on Zoom, you can put it on chat. And everybody who asks a question afterward has to use a microphone, because otherwise the people that are staring at the screen don't have any idea what we're talking about, and they can get quite testy about it. Okay, Todd Elliott. This is a local guy. He grew up right around here. He's been all over the world doing mushroom and plant and other things. His PhD research looked at the relationships between vertebrates and truffles, so that had to be good. He was formerly a professor at the University of New England, which oddly enough is in Australia. He's done a lot in the journal literature for you. I think we've got some really serious mushroom people in the room tonight, so you probably all know that. He's also the author of Mushrooms of the Southeast. I'm going to start one passing around so you can all look at it, and we have some available over here. So, and I said hold questions to the end, so turning it over to Todd.
How y'all doing this evening? Good. Well, I'm just thrilled to be here. So yeah, my name's Todd Elliott, and I'm passionate about all different aspects of the natural world. And I love the subtleties of interactions and how different organisms interface with each other. And I see fungi as sort of the great connectors. They're interwoven through many different aspects of our lives, whether we realize it or not. And 100% they're interwoven between all different organisms around us out in the forest. Before I get going, I know they said to hold questions to the end, but I will say if you've got some sort of theoretical question, we can hold that to the end. But if I get passionate, I get excited, I get talking fast, and if there's something you say, whoa, slow down, can you clarify that? Please do. I won't take offense to that, and I welcome it. So some of you in this room are probably, well, let's actually, I want to ask you all, how many of you feel comfortable identifying sort of 4 or 5 mushrooms or more? All right. And some of you, how many of you you feel like mushrooms are brand new? All right, a few of you. Well, before I get going, I want to start with just a little bit of fungal basics. You know, for many of us, if you go to high school and you take a basic biology class, you might get the basics of plants and how pollination works and those sort of basic concepts. But mushrooms are sort of a forgotten stepchild of botany. And so there's many sort of aspects of their very basic biology that many of us are totally unfamiliar with. And that's not a critique, that's just an observation. And I welcome that as an opportunity. We can all grow from that. So I want to start out by, anybody know what we have here on this first slide? Mycelium. So we have mycelium. So when you see a mushroom, that's like an apple off an apple tree. Most of the organism is spread out through the environment. And it's doing many different things. Sometimes it's a parasite, sometimes it's a decomposer, sometimes it's tied in with tree roots, and sometimes it's doing many different other things to the ecosystem. And we'll touch on a few of those as we progress through this slideshow. But to begin with, I just sort of want you to think about that. Now, when you see all of this white web spread out through the forest, that is hundreds or sometimes thousands of fine strands. And a single fungal filament, it's called a hypha, is stacked one cell on end, on end, on end. And you can't see that with the naked eye. But when you get a mass of them together, you get mycelium. And that's what you see here. All right, so how does fungi spread through the environment? There's a hint up here. Spores, look at y'all, you're really on top of this. So what color might this mushroom's spores be? White, how do you know that? Because it's a mycena. Charlotte's over here dusting off some fancy mycological terminology. That's a genus name for one group of mushrooms. But why else might we know it has a white spore print, aside from if we know that it's a mycena? Amen. Preach it, sister. This is because we have the white powder here. This is the white spores, and they're spread out through the environment. And I want you to think of those spores as sort of half of a seed. They got to spread out, and they land somewhere, and then they start to grow. And if all goes well, they'll find a compatible mating type, and they'll mate, and they'll fuse, and they'll spread out through the environment. So why should we care about these spores? Well, for one thing, they can be helpful for identification. And this talk is not going to focus on edibility, but learning to identify spores based on color is helpful whether you're interested in edibles or whether you're straight fungal taxonomy and you just want to learn how to identify them. So these two mushrooms look relatively similar, correct? The one on the left happens to be edible. The one on the right will make you mildly sick. What color does the spore on the left have? There's a hint. White spored. And the one on the right is green spored. So does that mean that all green spored mushrooms are edible or are toxic and all white spored mushrooms are edible? No, this is just a little piece of a puzzle that we can use to identify different mushrooms. So how would we know what color these mushroom spores are without putting them under a microscope? A spore print, exactly. So to make a spore print, we cut the cap off of a mushroom, we set it on a piece of paper, we cover it with something, and then those spores are discharged, and in the negative space, so the gaps between each of those gills, you'll see a spore deposit. It's a beautiful way to make art if you want. It's also really cool for identification. Now, here's a really cool little bit of fungal natural history. So if we look at the underside of a mushroom, there's many different structures, but these are the three most common structures. And what do we think the top one might be called? Gills. Look at y'all. I feel like I'm preaching to the choir. Gills, they radiate out like spokes from a wheel. And then if you look between those gills, do you see how there's some little gills that are short and some that are long? All right. If you're a technical mycologist, you would call the short ones lamellulae. Can you all say that? Lamellulae. All right, all right. And then if they're long ones, you call them lamellae. All right, those are just some fun words. Can we say lamellae? Lamellulae, lamellae. All right, we got this. And then some of them have teeth that hang down like icicles, and other ones have pores underneath. So why wouldn't mushrooms just produce one flat surface? Surface area, exactly. So some mushrooms do produce a single flat surface, but many of them choose these various types of contours. And it's really interesting because those features show up in many different fungal lineages, sometimes if they're not even related to each other. Okay, but now to go back to my earlier comment about how spores get out into the environment, what's really crazy is let's use a gill, for example, one of these blade-like structures. On the end of the blade-like structure, there's a little club. If you're a mycologist, you call this a basidia. On the end of that club, there's a little spike. And on the end of that spike, there's a spore. Now, fungal spores, they're roughly, it depends on the species, but they can be between sort of 3 or 5 microns up to maybe 20 or 30. And a micron, for those of you that aren't familiar, that's 1,000th of a millimeter. So tiny. Well, those little spores sit on the end. And as they mature, they sort of produce this little gas bubble and they build up pressure, build up pressure, and then there's an explosion. So those of you that have driven a car too fast, any of you? All right, or maybe ridden in a really fast roller coaster, you maybe experienced a G-force, that momentum that throws you forward of like 3 or 4. If you're an elite astronaut or a pilot, you might get a G-force of close to 10 or 12. But a spore leaving the gill, the G-force is close to 10,000. Isn't that mind-blowing to think about it? Now, the thing is, air has a lot of resistance. So that spore has to slow down before it smashes into the gill opposite it. And that's what the force it takes to punch through that air. And then they sort of bounce back and forth, back and forth, back and forth, back and forth. And then ideally, the cap of a mushroom has sort of roughly the shape of an airplane wing. And ideally, it sucks the spore out into the air. And spores, they're distributed widely. But what's really wild to think about is 80 to 90% of the spores land within about 6 feet of the parent mushroom. So that means when you're out there picking mushrooms and looking at them, you're actually helping with spore dispersal because you're moving them through the forest. All right, you all following? All right, so why do we see these shapes show up in out in the fields and forests? Fairies. I heard somebody say it. I'm glad we could get down to the point here. So yes, fairies. No, exactly. What actually happens is spores, once they land, they start growing out circularly. And so you get these wider rings. Now, I have a friend who's a mycologist and a pilot, and he's in Wyoming. And those are two professions that are pretty hard to meld. But what he started working on was fairy rings. And he found out in those plains, in his plane over the plains, he could see these circles that were expanding out and realized when he was on the ground, he might have only seen one mushroom here and one mushroom over there. But in fact, it was part of a huge, huge fairy ring. And in fact, you can all do this at home. He started trying to figure out how to age them. So if you have a fairy ring in your yard, you can mark it because they generally expand a little bit every year. And you could roughly calculate how old your particular fairy ring might be. But he came up with estimates on some of his fairy rings to be several hundred years old. So when we think about these mushrooms, these little ephemeral beings that sort of decompose after the fairies have danced in a circle, as we all know, that's the real explanation. They're actually very long-lived, many of them. And that's what, part of what they're doing in the landscape. Now, they don't all form perfect circles because there's environmental barriers. There might be a sidewalk. There might be a dozer. There might be other mushrooms because when they run into another mushroom, they get competition, just like plants do. All right, so everybody following so far? We doing all right? All right, so a little bit more of basic fungal biology before we jump into some of the different species. So how do fungi break it down? Well, they spread out through the environment with those little mycelial threads, those hyphae that spread out. And when we consume food, we eat it and we chew it up with our teeth, and then ideally our microbiome, if it's in good health, will help break it down and release those digestive things to our body. Well, fungi, they do it all externally. So it's not really great to do in polite dinner company, but you could spit in your meal and try to stick your fingers in it to absorb the nutrients back through. That's what fungi basically do. So they're exuding enzymes out into the environment, and those enzymes break down things, and then they suck it back in. And then those are basically how it feeds the fungus, or in the case of mycorrhizal mushrooms, they'll feed back into the plant network. But there's two main types. There's up top. Anybody want to guess what kind of decomposer this is called? Yeah, these are all right terms, but I'm drilling down a little even more precise. What kind of fungal decomposer is this? It's called a white rot, right? And then down here, what about this one? It's real technical. Brown cubicle rot. And you can see why, because it breaks it down in little squares. Now, the white rot, some of you maybe have been in a fine woodworking gallery and seen those spalting lines. That's what woodworkers call it. So when one fungus runs into another fungus, it's got two options. One is mate, or two is put up a barrier. And fungi, they use melanin. That's what makes those dark lines. So when fungi run into other fungi, they'll put up those lines. So you can work out the whole geopolitical status of a given tree stump by looking at those lines. And they're always having these territory wars and interactions between each other. And that's happening all through the forest. And it's easy to see it in wood, but it's less easy to observe in the soil. But keep in mind that's happening as different fungal communities run into each other. Now, the white rots break down mostly lignin. Now, I'm no chemist, but my chemist friends of mine tell me lignin is one of the harder organic things to break down. And so those are the fungi. Many of them are the ones that are desired for medicinal compounds. They're the ones that are producing really promising novel secondary metabolites and things like that, because they're pulling out all different things out of the wood or out of their whatever their substrate is that they're decomposing. And then the brown rots, they break down the cellulose and they leave behind the lignin. And the lignin, because it is so hard to break down, it's incredibly long lasting. And unless a forest is exposed to fire, lignin has been carbon dated in the soil to be sometimes over 200 years old. And it has surface area very similar to charcoal. And any of you that are interested in alternative agricultural approaches, maybe probably heard of biochar. This functions very similarly to how biochar functions in ecosystems. And for those of you that aren't familiar with biochar, basically you make charcoal and then you charge it. So you would say, for example, put it in a bucket with compost tea or with urine or something like that. It sequesters the nutrients, and it's not like a fast-releasing fertilizer that then you could put into your soil. So this will sequester it. It's hard for the plant roots to get it out of there. So they depend on their fungal symbionts to then mine it out of the charcoal, or in this case, out of the brown rotted wood. So it's wild to think about. Everybody following all right? Okay. So I want to touch a little bit more on some other types of fungal dispersal. And for those of you that haven't had a chance, I'm going to dip across a bunch of different fungi here. Anybody familiar with these? If you've ever walked by a mulch bed, you either have seen them or didn't notice them, one or the other, because these are one of the most common groups of fungi. Anybody know what they're called? Bird's nest fungi, exactly. So bird's nest fungi look like little nests. And these little packets here are actually spore packets. And if you pay attention, you see this leaf up here with my finger showing there for scale. Those are the spore packets. When raindrops hit, they'll eject those little packets out. I found those on a leaf close to 3 feet above where the little bird's nest fungi were fruiting. So they're actually exploding up, landing on the underside of leaves and sticking to them. It's a really fun thing to look for. Now there's other fungi, like anybody ever seen this one? All right, Charlotte over there. So this one is got a fancy Latin name, but the common name for it is cannonball fungus. Because it actually, these are immature, these little packets here, but these are about to explode, and they can shoot their little packets up to 6 feet in the air, and some estimates have been close to 15 or even maybe even 20 feet horizontally. And so they're just projecting these little spore packets out into the environment. And both these and the bird's nest fungi, some mycologists have argued that they're designed to look like seeds, and that way birds will eat them and then disperse them further, but who knows? All right, so then the fungi get out into the environment, and there's many different organisms that interact with them. Sometimes it's viruses, sometimes it's bacteria, sometimes it's other fungi. But they're mining the nutrients. Has anybody ever rolled over a log in the forest and seen these little translucent looking little millipedes? For many years, I just assumed that they were a juvenile instar, which is what a lot of invertebrate people call a younger non-adult stage of an invertebrate. But in fact, these are actually their own species of millipede, and the juvenile ones are these little tiny white ones. And they're mostly, the babies are mostly cared for by the father millipedes. And their diet is almost exclusively composed of fungal mycelium that's spreading through the wood. And what's really wild is the technical term that some of the millipede people use for this is they call it starring. You see how they're sort of all ringed in a circle? And that's because they all exude enzymes out of their mouths that decompose the fungal mycelium and the wood that has the mycelium, and then they jointly share drinking it back up. Isn't that wild to think about? And this is all happening in the forest. This is just a fun little tiny example of what's going on. All right, so how many of you have had that tree in your front yard that's got some shelf mushroom like this growing off of it? And you went, oh no, do I need to call the arborist? That tree might fall on my house. What am I gonna do? All right, I see a few nods out there. So there's fungi that specialize in decomposing the heartwood of trees. Anybody know what those are called? Cyprethites, but heart rot fungi. And the heart rot fungi, trees are mostly metabolically alive on the outer, say, inch or so, because that's where most of the nutrients are coming up and down. And that's not to say they're entirely metabolically dead in their heart, but they don't have as much going on. And so most trees really want to protect their hearts. I know some people like that too, you know? And they're trying to keep all of that material in the inside intact, because once fungi get in there, it'll decompose it and turn it hollow. Now, this isn't inherently bad. It's sometimes bad for the individual. But when you get heart rots, such as this Fulvifomes here, it decomposes the heartwood of trees and eventually will lead to cavities. So who's ever seen a pileated woodpecker? All right. And who's ever seen a gray squirrel? All right. These are common animals that probably unbeknownst to us, or to many of us, are actually very reliant on fungi in ecosystems. And part of it is woodpeckers, I've gone to stumps that they've chipped into, and I've taken my pocket knife and tried to chip into the same tree. They will chip through trees that are, you know, a formidable opponent for a stiff pocket knife. They basically have jackhammers for faces, and they will chew into the trees. But what's really interesting is sometimes they chip into a tree to forage on insects and things like that. But often when they're going into a tree, they're looking to make a cavity for their nestlings. So when they chip into a tree, they disproportionately, almost all woodpeckers, choose trees that have heart rot. And they can detect a tree that's live on the outside and has heart rot on the inside. They will chip in, and once they get through that outer living, probably a couple inches or so of wood, they'll create a hollow. Now, what's really unique about many woodpeckers is they have terrible, terrible nest hygiene. So if you're from rural Appalachia, you maybe have heard the name, the folk name for many woodpeckers. Anybody know it? Peckerwoods. And there's an expression that is familiar to many Appalachian loggers, is why that's as stanky as a peckerwood nest. And that refers to most birds carry their baby's poop out. But many woodpeckers, not all woodpeckers, but many of them do not practice this nest hygiene. So they will use a cavity for one season, and then they'll go on to the next place. Sometimes just around the same tree, they'll just drill another hole in, go to the same trouble. Now, this may seem sort of like extra work to you, and why would the woodpeckers do this? But it's actually why they do it. I don't know. You'd have to ask a woodpecker, and they're hard to be very articulate. They basically engineer the ecosystem to have all these other cavities for other animals. So very few birds, other than woodpeckers, actually will chip into trees. And then things like squirrels will come along and secondarily use their nests. And then other songbirds like bluebirds, they're cavity nesters, and many people put up boxes. But what's really fascinating to think about is many of the fungi that do the heart rot are actually releasing enzymes out of their mycelium that are cytotoxic. Anybody know what that means? Kills mites, or antibacterial, or antiviral. And so probably partly how woodpeckers can get away with such terrible nest hygiene is by having all these fungi in their nest cavities. And so there's this, and then many of the insects they feed on also secondarily disperse fungi because they're fungus beetles and things like that. So it's pretty mind-boggling to think about. Yes? Determine that it's rotted? All right, so the questions for those, if anybody's on Zoom, how do woodpeckers determine that a tree has been rotted? Well, there's some pretty controversial studies that they said they're using ultraviolet light and maybe that they can pick up through woodpecker vision. I suspect it could be olfactory cues, insect cues, and woodpeckers are always pecking on things, so they might actually auditorily hear it. Again, I haven't been able to ask a woodpecker, so it's really hard to know, but it seems to be very consistent. You seldom see any cavities that are not in a tree that has heart rot. And some people have done very complex genetic studies. Some people have just done observational studies, and it's disproportionately, even if you're just doing observation and not, because genetics, when they do, particularly with fancy genetics now, you can pick up every organism that might have, spit on it on its way by. You can get all sorts of diversity, but people who have just gone and inventoried the shelf mushrooms that are growing off of woodpecker trees have a pretty consistent list of species that are often associated with woodpeckers. So it's mind-boggling to think about. And the only places in the world that don't have woodpeckers, or large land masses that don't have woodpeckers, Australia, New Guinea, and New Zealand. And it's interesting, and they have very high parrot diversity. And so I'll keep this talk southeastern related, but here's one slide I'll just flick up to show you that this is a ubiquitous association globally. These are rainbow lorikeets in Australia, and you can see they're peeking out of the heart rot of a eucalyptus, and this is the punk fungus that causes those cavities, and the parrots are then pulling out pieces of brown rotted wood, just like we learned about earlier, and creating cavities. Isn't that wild to think about? So are all shelf mushrooms heart killers? No, not all of them. Many of them are. Some of them are butt rots. Guess what they decompose? The butt of the tree, the base of the tree. Some of them are root rots. Some of them are more superficial. Many of them are heart rots, but not all of them. Anyone who tells you one across the board of fungi is probably either simplifying or pulling your leg, because the fungi are too diverse to be fit into one box often. So here's a fungus I'm really fascinated about. It rarely makes it into anybody's top 100 favorite mushrooms, mostly because they probably never noticed it or realized why it's important. So you probably see this and say, wow, this is a really drab slide. I might take a quick little nap while he's on this one. I urge you to stay with me. This is a really fascinating fungus. This is called a glue crust. And what it actually does is it will stick sticks to other sticks and sometimes to living trees. And there's this whole group of fungi that actually will trap twigs in the canopy before they ever come to the forest floor, because competition among fungi is much more fierce down on the ground. And in fact, if they can hold it up in the canopy, that means they have a smaller range of fungi that they're actually competing with. And this one, right now I'm on the process of building a very slow-paced sculpture that I can just keep gluing twigs into it. But what's really fascinating is many birds will disproportionately choose twigs that are infected with this fungus. And they'll use them to weave into their nests because it will basically weld the nest to the tree. And then as those nestlings get fatter, it's actually holding them. And we'll show some more slides about this, but there's actually some studies in like the lowland tropics where they've shown birds that use these twigs had much higher success rate of nesting than ones that didn't, because again, the less were less likely to blow out of the tree because they were actually welded in place. So along that trend, there's another group of fungi. They're called litter trap fungi. So as you get into wetter forests, this becomes more common, but in the deep south, it's really common. In the mountains here, I find it pretty often. It generally needs to be in sort of a rhododendron thicket near creeks, and you'll see these often leaves dangling at weird angles. And at first you might go, oh, that's just a spider web or a caterpillar silk that's holding it in place. But then if you get a little closer, you'll see these black strands. I was actually given some long black strands from Watauga County one time by someone who thought they might have found Bigfoot hair. And it turned out that they were these marasmius rhizomorphs. So this is a group of little tiny fungi called marasmius. They look like pinwheels. You probably, they're the kind of fungus that's often showy but small, might show up as a passing, fleeting glimpse on your pre-programmed screensavers on your computer or something like that. But many of the species are seldom noticed. And actually, these are really vital in bird nests. I was really fascinated how widespread this was. And I came up with a list of, I think it was over 270 something bird species around the world that used these. And that is so far from complete, I can't even begin to calculate how off it is, because there's so many reports of people who were inventorying, ornithologists who were inventorying nests, and they'd say, oh, we found these mysterious horsehair-like things, but there was no horses around, and they had a white interior, and we assume they must have been horsehair. It's very obvious that it was actually this fungus. And some of these have never been known to fruit, so they seem to be entirely dispersed by birds who weave them into their nests. And so then it's moving it through the canopy. And again, these are the litter trappers that are decomposing, which is important here as you move into the actual lowland tropics. Many of you probably heard those stats about how much of the life activity is happening in the canopy and how little of it's actually on the forest floor. And a lot of that decomposition is happening on account of these fungi. Remember what I said earlier about melanin? So fungi produce melanin to protect themselves when they're in harsh environments. And though the canopy of a forest may not seem that harsh, but if you're a fungus, one of the things that you're fighting most against is drying out. And so by coating your threads in that melanin, that makes those, they're called rhizomorphs. They make the rhizomorphs because rhizome meaning like root and morph meaning like the shape. So it literally means root forms. And it basically protects them in the canopy. So y'all following? So just to cap this, I'll just talk briefly. This is a bird's nest I found down where I live in Rutherford County, North Carolina. And you can see the rhizomorph all the way over to my finger. And the bird has worked it into the nest here. It's many of these black strands. But then those going up along that edge are actually, in fact, regrown and attached to the twig. Isn't that wild to think about? So I urge you, look at bird nests differently. All right, so a talk about mushrooms is incomplete without at least mentioning lichens. Lichens are on the margin of what my deeper understandings of fungi are. So I won't spend too much time on them, but I want to point out to you that lichens are incredibly diverse. And in fact, they're a very hard group of organisms to classify, because if you have an algae, which is part of the association in a lichen, and then you have often what's called an ascomycete. So like the most famous ascomycetes would be morels. So there's an algae, and then there's an ascomycete, and then there's often a cyanobacteria, and sometimes now we know a yeast as well associated. And then occasionally you can get basidiomycetes, which is like this lichen up here on the far right, those little clubs. They're related to coral mushrooms, which is the same group of mushrooms that has your amanitas and your chanterelles and your bolete. That's called the basidiomycetes. And you'll find the one on the upper right, only growing on logs that are covered in algae like that. And in some parts of the world, there's also a couple species that will grow on soil that's covered in algae. So in fact, they are a type of lichen. And lichens are really important for both air quality. They will decompose, they can mine rocks. They're slowly breaking down nutrients out of rocks. They're slowly breaking it down out of wood. They perform many, many, many different functions. They're really interesting for dyes. Some of them have some toxic compounds in them. This is sometimes called like a wolf poison lichen because apparently they'd mix it with meat in the old days, put it out to bait wolves and could theoretically kill them. I haven't tried this because I hear it's not the most appropriate thing to do, killing wolves at helter-skelter these days. But back in the day, supposedly that's what was done. So another thing that I want to break down for you all, as fungi do in our ecosystem, they break down lots of things, is mycorrhizae. So what might that word mean? If you're literate, you might be able to help me out here. Myco means fungus, and rhizae means root. So it literally means fungus root. So you probably heard that term get beat around a lot these days. It's the buzz in agriculture, it's the buzz in forestry, and we're beginning to realize how integral that is to how forest systems function around the world. So when you plant a bean or something like that in a jar and you see that first roots come out, you probably see these little hairs come off the roots. So those are called, anybody know? Well, these aren't mycorrhizae yet. These are called root hairs. There you go, hairy roots. So most of the plant actually wants to get a fungal connection, or what's called mycorrhizal connection. And if you look at this upper left slide, those are pine roots. And you see how many of them are sort of swollen, look like maybe little, sometimes in books they're described as little balloon animals, little twisted, swollen roots. And those are actually where a fungus has grown around a root. And they increase a plant's area of absorption by hundreds, if not thousands of times. So underneath your boot print, you could have close to several hundred miles of those fungal filaments spread out. And they're actually tied in with the roots, and they're increasing the plant's area of absorption mind-bogglingly large amounts. So the plants, they photosynthesize. What's one of the main byproducts of photosynthesis? Sugars. And sugar, you can sort of think of it as the currency of the natural world. The plants are trading the sugars for phosphorus, water, nitrogen, the list goes on, that fungi are particularly adept at pulling out of the environment. So this exchange goes back and forth between the two groups of organisms. And sometimes if you're really lucky, you can see like these are what baby mushrooms are called? Primordia. So these little primordia, this is a full-grown adult mushroom. And you can see the roots associated with it. You seldom see them that closely attached, but it does happen on occasion. So those are spread out through the landscape. Now, I've heard this a lot, and this is something that you read in many ecological papers and things talking about fungi and their importance in landscapes. But one of my mentors, who I was very fortunate to get to spend many years studying with, is a man by the name of James Trappe. He published his first peer-reviewed papers in the 1950s and was sort of one of the pioneers of mycorrhizal research. And he did a study in the 1960s that sort of, in many ways, I think it's one of the sort of poster child examples of how important mycorrhizae are. And I'm just going to flip through a few quick slides to show you. So, excuse these photos. These are from the 1960s. And so Jim Trappe, there was a Forest Service experiment station in Washington State that decided they were going to do a Doug fir plantation. And in the Doug fir plantation, they got some reclaimed potato farming land. Now, the Forest Service in those days, they had all sorts of wacky ideas. But one of them was that potatoes would carry a huge amount of pathogens. Now, potatoes can carry pathogens, but they're mostly problematic for potatoes and other solanaceous plants, not for Doug fir trees. But they said, these potatoes are going to carry all sorts of things. So we need to sterilize the soil. So they said, well, potatoes are bad, so maybe let's use 3 times the recommended dose of methyl bromide. Now, those of you that aren't chemists, methyl bromide, that's what they gassed people with in the trenches in the World War. Except when they did it in the World War, they didn't mix chloropicrin with it. Chloropicrin's a vomiting gas that it acts the same as methyl bromide, and if you inhale it, you start puking, so then you know you inhaled methyl bromide and you might die. So they did 3 times the dose of methyl bromide. This is really scary stuff. And then they planted their Doug fir. Now, if any of you happen to be Doug fir farmers, please feel free to chime in. But most of my Doug fir farming friends say that in about 6 months, you should have seedlings that are close to this height. These seedlings are close to a year old. And basically what they did was sterilize the soil. So then they said, well, let's try fertilizer. That must be the problem. So then they added fertilizer plots across it. In some places, they tripled the recommended fertilizer dose. Again, nothing really happened. So then about a year plus on, they called my buddy Jim Trappe in because they said, maybe it has something to do with mycorrhizae. So he started looking across their plantation, and every now and then he'd see, like, do you see that seedling off to the left of the pencil, on the business end of the pencil there? You see how it's twice the size of the ones to the right. And then there's a little clump over here where the pencil's standing up. Notice how much taller those are. Well, he started pulling some of those up, and he'd notice that there was colonization. There was mycorrhizal root ends on there. And here you can see there's these random patches of taller seedlings in the mix. Oops. And these lines are their fertilizer plots. You can see there's no correlation between fertilizer loads because some of them go right across it. So what he realized was happening, and then about 2 years on, he started finding these are early successional species of mushrooms. This is an anosebe for those of you mushroom nuts out there, or sometimes affectionately called I don't noticeabees because they're really hard to tell apart. So this is one of the anosebes that sprouted up and was one of the early successional species that came in. So then the Forest Service, after a little while longer, they ended up with this really irregular plot. See how some of these have barely any colonization, and some of them are 3+ foot tall. So they had to turn the whole operation under. And then when they planted it next year, they all came up at even rates because the mycorrhizal fungi had recolonized those beds around them. So I really like this study because it really brings it home the importance of these connections. And in fact, most of the 80 to 90% of the plants around us wouldn't be growing as we see them grow today if it wasn't for their fungal symbionts. It's pretty wild to think about. How folks feeling? Staying with me? All right. So we talked about mycorrhizae. Now, very often this plant, and there's several others that look like it, that get brought to me, people saying, oh, is this a mushroom? Well, no, it's not a mushroom, but a mushroom is very integral to its natural history. So if you were to pull one up, I don't recommend you pull one up, you would see this sort of what's called almost like a tuberculate mycorrhizae, basically a clump of roots that a fungus has grown in between. And this fungus is what, or this mushroom, this plant is what is called a mycoheterotroph. Okay, this is a big word, but for a very integral concept about how fungi are the great connectors. So mycoheterotrophs, what they do, they don't have to photosynthesize anymore because the simplified explanation is they're a parasite on that mycorrhizal symbiosis we talked about. That's probably a very, very simplified version because the more we study the natural world, the more we realize that things we think are parasites are actually contributing something to many of the exchanges. But how this was explained to me is basically you have a plant photosynthesizing, it's producing the sugars, and the fungus is giving the plant. So all of those nutrients that are required for a plant to grow are being passed back and forth from one end to the other of that exchange. Do you follow? And then this fungus, this plant comes along in the middle of that exchange and takes nutrients from both ends. Well, what's really interesting is if you look at these plants under a microscope, they have chloroplasts, but they're not green and they don't function like chloroplasts normally do. So they're basically the parasite on the symbiosis, but they're probably actually contributing nutrients back into that exchange as well. And those of you that are into herbal medicine, this plant has gotten a lot of attention in recent years for dealing with trauma and dealing with sort of energy issues. And I know some people who work in settings where they're trying to deal with people who have been exposed to mind-altering substances and are really out of balance, or people who are dealing with really deep, dark trauma in their lives. And they'll say that like a small drop of this has been really helpful. And I've been in recent years hearing a lot of people talk about that. I'm not a practitioner, but I've heard some very compelling stories about how it's worked for some people. But dosage is really important. All right, so since we're not talking about human consumption of fungi in today's talk, I thought we'd talk a little bit about wildlife consumption of fungi. I generally steer away from slides that have a lot of text on them. I try to stick to photos, so that way if you're zoning out, you at least have something pretty to look at. But bear with me for a minute. So we talked about how trees are mycorrhizal. And we talked a little bit earlier about how fungi forcibly discharge those spores, those explosions that take those spores out into the air around them. But remember how we discussed 80 to 90% of the spores land within 6 to 8 feet of the parent mushroom. And you think about it, that's not that efficient for dispersal. So many fungi have developed a mechanism to combat that. And that is by forming sequestrate or truffle-like fruiting bodies that are under the ground, and then they can control when they're picked. Because if you're a mushroom sticking up above ground, any old animal that sees you can chew you up and eat you before your spores are mature. But if you're underground, you can release very specific aromas that will attract animals to come and eat you. And then many spores, actually, their germination rates improve after passage through an animal's gut. So you get things like these sort of associations that are going on. And then the trees, it's integrally important to them because they're getting new fungi passed through. And fungi mate all through their lives. So they're getting new genetic input. So they can adapt. And animals as their vectors are one of the really important ways for those fungi to be adapting to changing climate, changing humans' impacts on landscapes, all those sorts of things. So right here in the southeast, who's seen this mycophagist? Very slow pace, but it gets the job done. And you think about it, a box turtle, their home range is, sometimes less than a quarter mile, less than a half mile. But yet, most of those spores are landing in close proximity to the parent mushroom. So even a box turtle is significantly impacting those fungi's ability to move through the landscape. And what's really interesting is these box turtles, they are incredibly omnivorous. They eat all different types of mushrooms. The top two, they're probably eating the same species of bolete. This is a lactarius, a milk mushroom, a hygrophoroides. This is an amanita. And do box turtles eat toxic mushrooms? Absolutely. And there's actually interesting stories about the box turtle and human poisonings, because back in the day, a lot more box turtles were eaten. And there's actually accounts of people getting poisoned by box turtles. And some leading herpetologists of their time claimed that it was on account of the box turtles eating fungi. Now, that were toxic. And I have dug through the literature. I found lots of reports of that. But what's really weird is all of the other turtles, terrestrial turtles that are consumed around the world, in Asia, in Africa, in South America, I can find no accounts of poisonings, even though many of them eat toxic mushrooms as well. So I don't know if it's quite that simple. And unfortunately for the box turtle, not that many box turtles get eaten anymore. But the last sort of large-scale poisoning was during the coal mine strikes in Scranton, Pennsylvania in the early 1900s. The good old coal company clamped down on people's rations because they wanted, a living wage. And so they were starving them out to try to get them to work back in the mines. And many of them ate a lot of box turtles to try to get by. And there was a bunch of them that got poisoned. So who knows? They were probably also eating other things they shouldn't have been. But then there's a lot of other scattered reports through the literature about that. So it's a fun or an interesting thing to think about. Okay, but then there's lots of other organisms that feed on fungi, including probably some of you in this room. And what's also fascinating is many of the organisms prioritize the reproductive surfaces of the fungi. And that's because not all parts of a fungus is made nutritionally equal. So you'll often see where slugs have totally obliterated the underside and all the spores are being produced on that part. Or if you look at that uppermost slide at A on the left side, that you can see those are called eyelash cups because you see those little black hairs along the edge. Aren't they cute? That slug is coming along and it's eating all of the inside of that cup because that's where the spores are carried. And what's also fascinating is not all spore dispersers are made equal. So if you look at the passage rate of a mouse to a pig, a mouse will scarify the spores less than a pig will. So therefore, it doesn't mean that the mouse's digested spores are less viable. It just means that they're less scarified and germinate at a lower rate than the pigs do. Because the pig, if you put it under a scanning electron microscope, it will actually basically scratch off a lot of the ornamentation on the spores. And then you get things like these are springtails. Springtails are a little small invertebrate, and you see that brown streak down the middle of its body? That's its digestive tract. And it's just filled with the inside part of this truffle. So that can sort of show you how truffles, in many ways, they don't waste all that reproductive tissue on a stem, which has no spore-bearing surface. They're all basically one glob of spores sitting under the ground. And then vertebrates do similar things. So like the upper two slides, that shows the same, that's one of my favorite edible mushrooms. That's a leatherback lactarius or leatherback lactifluus now, if you keep up with the taxonomy changes. And you can see the top of the cap has had a few little chews out of it, but they've just eaten all the gills off the bottom. And the same sort of thing going along. But then you get things like puffballs, where they just sort of took bites out of them, and these spores weren't even mature yet. But these amanitas, the caps are gone. And also, you know how trees will have a browse line where you might have to make sure your goats aren't eating up too high. That's why you keep your branches. Deer will do the same thing with mushrooms. These are called ichnodermas. And you can see the browse line all the way up to about head height, as far as a deer could reach. So wild to think about that. So the Southeast is home to many different types of truffles, and many of these truffles are overlooked because they tend to be under the soil, under the leaf litter, because they're termed as hypogeous fungi, meaning under the ground. And many of them are very colorful. They're like little geodes. Most of them are very small, but they're all designed for animal dispersal. And whether you believe in God's handiwork or Darwin's handiwork or evolution's handiwork, it's a fascinating and really beautifully well adapted aspect to the landscape. Here's some different truffles. All of these could be found within close proximity to here. And one of the most common groups is the Elaphomyces, or the deer truffles, called that because deer love to eat them, as do squirrels and other things. And what's really fun to look for is very, very often I'll find the same kind of locations where a squirrel will have sat and husked acorns or hickory nuts, and you'll see this little bit of black powder scattered across there. And that black powder is actually, if you put it under a microscope, will match some of these different species of southeastern Elaphomyces. And if you don't have a microscope, you can actually pick up the spores and rub them between your fingers. And they feel a little bit like chalky velvet. That's the best way I know to describe it. And that's a good indicator. But if you put them under the microscope, I'll show you a fun little. Oh, before we get to that, I'll touch a little bit more on squirrels, and particularly red squirrels. They cache fungi, and they love to particularly cache deer truffles. So here's a robin's nest that was found, and it was full of that many little deer truffles. You can see all packed into that robin nest. It's just wild. Looked like a Photoshop job, but it really was. And then other red squirrels, particularly as you get further west or colder, will actually hang them out to dry. And what's really interesting is there's some amazing and wonderful behavioral studies of people who sat and watched squirrels from the first days they came out of the nest. And they talked about how it would take baby squirrels sometimes a week to be able to hang and dry 1 or 2 mushrooms, whereas their mom could hang 100 in an afternoon. Because they usually choose forked sticks, and then they'll set them through there with the stem and the cap. And very often, the squirrels will struggle with it, and they'll break them, and they'll run back and forth down the trees. And if you've ever spent any time with red squirrels, you know they're very talkative about everything they do. And the harder they struggle with hanging the mushrooms, usually the angrier and louder they get. But that also, remember we talked about spore dispersal. They're hanging those mushrooms way up in the canopy, basically making a spore print, like we did on our kitchen table when we're trying to identify a mushroom, which gives an incredible ability for those spores to be dispersed all through the air, but also the squirrels are eating them, and then their little poops are getting dispersed widely across the landscape. So it's a wonderful association between the two organisms. And also, many larger animals use fungi as sort of their medicine cabinet, because they get their multivitamins by eating a few fungi. There's a lot of nutrients that are really hard for organisms to get out of plants. So even things like polar bears, deer, buffalo, elk, things like that, they're not going to eat a large volume of fungi relative to what their normal pound of food intake is per day, but they'll add a little bit in, and that's giving them nutritional compounds like selenium. It's a really hard organic compound for fungi to be able, or for animals to be able to get, but fungi produce relatively high levels of it. So it's really important for wildlife nutrition, but also many of the small animals will only have to go drink water because fungi are high enough in water that they can avoid that exposure to predators by just eating enough fungi. How many of you have had a pack rat do damage to your vehicles? All right. I see a couple of you. Where I'm from, one of my local mechanics, that's part of how he keeps in business, is all the wiring that gets chewed by pack rats. And the main pack rat we have in this area is a bit endangered. So it may depend on where you're living. And if some of you are closer to actual proximity, you may not have as many of them around. But they hoard objects and they dry objects. And this is a friend of mine who does work in cave systems in Ohio. She sent me this picture. Look at this bouquet of leaves that the pack rat stored, along with lots of russulas and lactarius that it's dried and hauled into this cave that then it's going to continue to feed on over the winter. So it's a really important aspect of food for the animals in the landscape. But what's also really interesting is red squirrels. Those of you that are familiar with them, they're incredibly territorial, and they'll actually sometimes fight to the death over cache ownership. But what's interesting is they're diurnal, whereas flying squirrels are nocturnal. And in some parts of the world, flying squirrels get their way through the surviving long winters by raiding red squirrel caches at night while the little raging tyrants are sleeping. So it's really funny to think about that going on. And sometimes even jays and other, like particularly when you get further north, Like the Siberian jays, they eat a lot of red squirrel or European red squirrel cached fungi. And that's partly how they get through the winter, is by feeding on the red squirrel caches. So that's partly why the same species of squirrels, caching levels depend on the climate. So the same species in, say, like Scotland will cache way less than the species in Finland or Norway, because their winters are so much drier but filled with snow, whereas further south it's wetter, so they can get food that they buried. Whereas once you get a deep pile of snow, you're not going to be able to dig up your buried nuts and cones and things like that. So you have to have arboreal caches that you can get through. So yeah, it's fascinating to think about caching. While we're talking about the squirrels and how the black powder, you see on this, those of you that are into your boletes, this is Boletus hortonii, and you see that black powder on there. That's actually squirrel backwash from eating a deer truffle. So I took that back, put it under the microscope, and looky there. Those are what the Elaphomyces spores look like, and they were sitting on the cap, and the squirrel was so excited. It just finished an Elaphomyces, and then it jumped onto the bolete, and it still had some truffle spores hanging in its mouth, and it literally backwashed. It looks like a Photoshop job. I promise you, I did not alter this image. So it's wild to think about. All right, we got a few more minutes, right? Yeah, okay. I want to touch on a couple other bits of these sort of fascinating interactions. Let's push a little bit more towards the invertebrate world. So probably most of you have never noticed this fungus, but how many of you have noticed a black gum tree? All right, a few of you, or at least know what a black gum tree is or heard of a black gum. So this fungus is so common on black gums, you could practically use it as an identifying character. Not every black gum, but most black gums have it on there. And what's really fascinating is it looks like this little bit of like creosote on the branch. But in fact, if you peeled that creosote back, it's like a little bit of a felt layer that covers a community of scale insects. And those of you that know scale insects, they have a proboscis. So like butterflies have those beautiful little proboscis they roll up. Mosquitoes have that little that stick in you. And that's also a proboscis. But the scale insect proboscis is stuck into the vascular system of the plant, and they're sucking sugars out of it. And what this fungus will do is come along, cover that community of scale insects, because many scale insects, particularly the females, are flightless. And they'll sit on that same twig, sucking out the sugars from the tree. But this fungus will come along and it will parasitize a few members of the community, keep their exoskeleton intact, and drink sugars through that scale insect's proboscis. But it has a net positive for the whole community of scale insects, because many of them are protected by this covering. So it's kind of like who draw the short straw, you know, in that scale insect community, because a few of them will die, but then the whole community, it seems like, benefit. And this is a really diverse group. There's, I think, close to 300 described species in the genus. Many of them are specific to different trees. If you really want to go down that rabbit hole, there's a 500-page monograph that has about 2 pages of natural history, and the rest of it is keys between the species. Yes? That is a great question. So for anyone on Zoom, the question is basically, are the scale insects specific to the specific tree and the specific fungus? So probably yes. There's so little known about this group of insects and fungi associates that probably many different things are happening. But my understanding is it's currently about a species of Septibasidium per plant species, and some of the plants are so divergent. There's probably some overlap, but there's like brown Septibasidiums that you'll see on oaks and citrus and white pines, and they all have different host trees, and they're probably many of them separate scale insects. But the problem is finding someone who's both a good scale insect taxonomist and a good Septibasidium taxonomist. That's a really rare combination. If you know any, let me know. I got lots of questions for them. So I want to talk a little bit about zombie insects or insect pathogens. These have gotten a lot of attention recently. I've had lots of people ask me, have you seen The Last of Us? I haven't seen The Last of Us, but I get the idea of the premise. And that's a organism that takes over the bodies of other organisms and manipulates their behavior. And that's absolutely happening in the fungal world. Whether it's happening with zombies and humans in The Last of Us, that's to be seen. But so far, I haven't seen any compelling evidence of it yet. But what's really fascinating is the Southeast is one of the most diverse places to find these insect pathogens in the temperate world. You move into the tropics and it's a whole other level of diversity. But within Western North Carolina, there's easily 40 to 50 morpho species. So things that are easy to tell apart without even getting under a microscope. And then if you delve into doing genetics or microscopes, there's a whole lot of other undescribed species. This one here on the cricket, it only parasitizes Carolina leaf roller crickets. I found it on the UNCA campus. It's still an undescribed species. No one's put a name on it yet. These two, this species and the other top one, are both ant parasites. Those were only described in 2017. So there's many, many still to be described species in this group. That one on the top is Ophiocordyceps kimflemingi, named after Kim Fleming, who's done had a deep passion for insect pathogens and lives in upstate South Carolina. And there's many different ones. Some of them, this is an asexual stage. This is a genus called Bravaria, and it's incredibly omnivorous. So it eats, you'll find it on everything from praying mantises to scale insects to beetles to bees. And some of these have potential as myco-insecticides, so used in a way instead of insecticides. Now, there's always controversy around deploying any sort of biocontrols and what's going to happen. Right now, we use large-scale, non-specialized insecticides and blow them across the landscape in large levels. That's also controversial in my mind, but we widely do it. Can we use these in a safe way? Probably. But there's still a lot of research. Some parts of the world, they're actively using it. This is a great one to look for. It's pretty common this time of year. You'll see those bright golden heads sprouting up out of the ground, and they only grow on bees and wasps and hornets and that group of insects. So fascinating. That one on the top middle, that's on a cicada. The other one, really common, grows on moths. And then this one has gotten a lot of media attention because of its use in medicinal preparations. Cordyceps militaris. It's probably one of our, as far as biomass goes, one of the larger species that you're going to find in the Southeast abundantly. You can find areas where you can find dozens in an afternoon. And so there's been a lot of research because out of Asia, there was a cordyceps that was widely used in medicine. And it got a lot of attention because I think it was a 95 women's Olympic team, broke all these records, and the coach swore that there was no doping and that the only thing that the coach gave them was the high Himalayan cordyceps. It turned out there was also doping, but this fungus does do the similar kind of things where it can boost white blood cell count. It's good and has potential in dealing with cancer treatments. Now, again, there's a lot of bad science and there's a lot of good science in medicinal mushroom stuff. So you'll find a lot of people that fall very firmly on one side or the other of that divide. And you can absolutely find really bad studies, and you can also absolutely find really compelling and interesting studies. And many different compounds, such as cyclosporin, the main immunosuppressant that was first enabled organ transplants, was derived out of this cordyceps that grows on insects. So this isn't just for any of you that are super skeptical, this isn't all just some far-flung folk medicine thing. There's a lot of actual clinical stuff that's actually been extracted and is implemented today. Early stage antibiotics being one of the obvious ones. So how much more time we got? 5 minutes? All right. Well, I'll just wrap up with talking about this really interesting association. This is one of the false turkey tails. And this is one of my favorite ones to look for because I seldom see this fungus. But what's really fascinating is the backstory behind this fungus. So I've still actively looked for many years to try to find this pigeon trimex wasp, and I have yet to actually see it in the wild. I had to go to an entomological collection to get this picture of it. But what's really interesting is this wasp carries that fungus in her, basically she has a spore-carrying compartment that she carries an inoculum. When she goes and oviposits, which is what insects do when they lay eggs, so it oviposits in a log, that fungus is mycelium. And that mycelium grows through the log, and its larvae feed off of that mycelium. Okay, cool story, but there's a whole other level. Have any of you gotten to see the giant ichneumon wasp? All right, I heard someone say, oh yeah. So this wasp, you can see an incredibly long, anybody want to guess what this long, looks like hypodermic needle or sewing thread is called? An ovipositor, basically designed for laying eggs. And then on either side of it, those sort of squirrely little curly cues things, those are sort of like part of a drilling rig. So I've gotten to witness this a few times. So what she does is she uses her feet. She can detect where the pigeon trimax larvae are and where they're feeding on inside of the log. She'll detect where they are, then she'll bend her body at that J formation. She'll plant those drilling rigs, and then she'll drill sometimes 3-4 inches down into the log to then oviposit her eggs on the larva of the pigeon trimax that is feeding on the fungus that it's inoculated all in the log. Ain't nature grand? It's just a wild, wild association that's happening like right under the bark. And one time I've watched it happen 3 different times. One time it was on a log. I watched the whole process and was like, wow. And I got up to walk away. And then there was another one on the same log. So if you find one, it's worth hanging out and trying to go back and keep an eye on it because it's just a fascinating interaction. And you'll get to watch. You can sort of see this, like, my understanding is there's a way to pressurize it, this part of her abdomen that fills up when she drills down in and lays the eggs in there. So I'll point out on the last minute or so, this is another really fun one to look for in the forest. It grows on beech trees. And this time of year is a great time to see it because you can actually see up in the upper left, the beech blight aphid. And these beech blight aphids, when you walk up to them, they all start dancing. And what's really wild is if you stick your hand or your arm underneath where they're dancing, you'll notice and you'll start to feel after a moment or two, these little droplets hitting you. And those little droplets are actually sugar excretions. And I kid you not, you can lick the sugar excretions off your hand and they taste sugary. And then there's a fungus that comes along, this one here. When it's first, this time of year, it's generally more white colored. If you see it in the winter, it'll look like a piece of char cloth hanging in a tree or on the ground. And you'll see these sugar deposits where it's feeding on the fungus. The fungus is feeding on the sugar produced by the aphids. And then at other times, you'll even see sometimes yellow jackets and butterflies and birds coming along and drinking sugar off of those piles that the fungus is then sequestering, and then the insects that are coming along. So it's just mind-boggling to think about. So, all right, well, I think I'll wrap up there. I want to thank you all and some of my funders for various aspects of my research. And anybody got any questions? Yes. Is there anybody on Zoom? Okay. No questions from Zoom. Okay. So let's see. I'll run this mic to you. Who was it that had the question? Oh, you got another mic too. All right, here, I can use this one for now.
So getting back to the heart rot fungus, does the fungus try and limit itself to make sure that the tree stays alive, or does it completely end up usually killing the tree and end up making snags for the for other wildlife?
Yeah, that's a great question. And probably some of it's both is happening. For the most part, many of the trees that are just heart rot have heart rot fungi in them. They can last for a really long time. And usually what actually ends up killing them is they won't quite have the structural integrity of it to sustain another tree hitting them, or a big wind event or a big snow event or something like that. They also, just like us, at a certain point in our lives, we become more susceptible to various pathogens. And so that may be the point, because all trees don't live forever. So that's sort of often an indicator that they're getting towards the later part of their life and playing out a very vital part of the landscape. That answer your question? Yeah, no problem. Thank you. Other questions? Yes, you want to run that mic back?
So are there any known Septobascidium species that interact with the woolly adelgid on the hemlock trees here?
Great question. I have not heard of any of them that do, and my understanding is it's a different group of scale insects, but I will be very upfront that I'm not a scale insect taxonomist, so I can't answer that question fully, but I don't know of any that do. Yes, I think, did you have first?
I wanted to know how you find the truffles.
How you find the truffles? That's a great question. If I told you I had to hide your body in the leaves. No. Look for animal cues. That's the #1 thing to do. So you can use an animal. So you can train a dog or a pig or something like that. Most pigs you don't have to, and it's hard to subtly go truffle hunting with a pig. I'm gonna take my pig for a walk. But if you look for where an animal has dug, you'll often see they will generally, because many animals want to balance their fungal diet. So they don't pig out on one truffle usually. So like the California redback vole, small little vole, it's been reported to have 8 to 10 different genera of truffles in its gut at any given time. And that's because fungi don't have a complete set of amino acids. They carry all the amino acids we need, but not all in one species. So they'll take a few bites of this one, a few bites of this one, a few bites of this one. So you can safely assume that when you see a dig, there's probably other truffles there. And also, they are only able to pick up truffles by smell. So they're not going to be thinking critically in the same way we would have, like, oh, this is the right kind of habitat. This looks similar. There might be some here. Because I've been out with highly trained truffle dogs, seen a truffle bulging out of the ground and drug the dog back to it. And the dog would step on it and then signal to one 6 inches away that I totally missed because that one wasn't mature and wasn't releasing aroma yet. So if you're willing to dig, you'll often find them. And like many of the truffle biologists will sample by just using rakes, and they'll just rake in the suitable habitat and then roll the leaves back over. And you can find a lot of diversity that way. Does that answer? Question? So most truffles land within sort of the upper humic layer, and you can pull back the leaves and they'll be sort of in that sitting on the top of the soil. And then a few species will actually be down in like technically the mineral soil, but less diverse there.
So to piggyback off the truffle question, no pun intended,
Sorry, go ahead.
I've heard that pigs carry a steroid or called adenosine, I think it is, and it's also found in truffles, and that's how they're able to kind of find them. Is there any truth to that, or is that just something that's alleged?
So that's a great question, and there's a lot of controversy back and forth about that. There's some people who claim it's all patriarchal science that's just trying to say that only female pigs are receptive to truffles because it mimics the smell of a horny boar hog. Now, whether that's true or not, I've, again, not been able to sit down with a pig and had a real heart-to-heart about it. But my understanding is that they're so motivated by the food and nutritional value of it, they're just able to pick up that whole profile. Now, when we see with dogs, the ones that have been trained on white truffles versus black truffles, that's taxonomically a whole different set of species, but their aromatic profiles only differ in, I think it's like 1 or 2 atoms. And the truffle dogs that are trained on one species are far less effective than ones on the other species. But then some people, just like you can train drug dogs to be much more generalist or specific, some people have trained their dogs to be broader spectrum. But dogs that are like only focused on specific species and specific cultivation operations don't. But what we know with like orchid pollination and how there's all this trickery going on, it's very likely going on, but most of mycological research is like 100 years behind botany. So we hope to be finding that stuff out soon. So it's a great question. And I think it's probably not quite as simplified as much of the media has done, but there's probably some truth to some elements of that.
Okay, this is a super basic question.
I love
'cause
them.
we're
Makes it
getting to
easier.
mushrooms, but so like ramps, there's ethical ways to forage and collect that. Is there like a basic level of ethic with certain mushrooms or certain species that like we should be abiding by when we're out in the forest?
Great question. So for the most part, that's species specific. So any of the mushrooms that are mycorrhizal or short-term in their growth patterns, you can, as long as you're picking, either leaving some that are mature or you're only picking mature ones, you can pick all you want. And the only people you're going to inconvenience are your competitive foragers that are going to go along after you. For things like chaga and some of the perennial mushrooms, that depends on where you are. So chaga in the Southern Appalachians, we're on the southernmost tip of its range. And the ratio of chaga to, particularly in the Asheville area, to people interested in chaga is not sustainable. If you go to the northern Midwest or Northeast or Canada, you could strip the forest. There's like logging sites that are cutting down, and you could go in behind the logging trucks and pick huge amounts of chaga. So a lot of people say chaga can't be harvested sustainably. That's mostly a conversation relevant to the Southern Appalachians. When you get further north, it's really quite common. Out west, the matsutake, people rake for it, and the Forest Service has actually put in regulations on the size of the buttons you pick, because there's, again, it's so valuable that a lot of people will go and they commit what's called rake rape, where they just go through the forest and it looks like feral pigs have come through, and they'll tear up large sections of the landscape. Fortunately, we don't have anything that's going on quite like that in the Southeast. So those questions are less relevant to here. Also, things like reishi mushrooms. If you're picking the hemlock reishi, that's an annual mushroom. But some of the really big polypores, they're 40, 50 years old, some of them. Now, that's not to say many of them are common, but the same way you'd think twice before you cut down an old growth oak tree, you want to think the same way. Not saying that you're going to deplete it, but just maybe don't pick that one unless you really have a use for it, kind of thing. Does that answer your question? Okay. You have one up here?
So for arboreal mycelium, that some of the cool ones you showed, what is their relationship between arboreal lichens and other things? So is there a lot of competition there? Is there cooperation?
Yeah, so great question. I think it depends on the niche. So for a lot of lichens, they have to have a fair bit of light exposure. And the niche that a lot of the sort of litter trap fungi are occupying is totally different. So not to say that there isn't some competition going on. I haven't seen any obvious trends of that. Like when things get on the forest floor, you get sort of the fungi that grow on bare dirt are obviously interacting with when there's litter deposits and those sorts of things. But in the canopy, I haven't seen it explicitly studied. And from what I've seen, there's not been a lot going on, but that doesn't mean it's not happening somewhere. Other questions, comments, additions, tangents? All right. Thank you all so much.
Well, this is a fascinating talk, and I'd like to tell you that one of the wonderful things about being here at Ollie, and we do appreciate so much the opportunity to do this, is that we can Zoom it to people who don't attend, and we can record it. And if you hunt around and find the website of the Blue Ridge Naturalist Network, Blue Ridge is 2 words, we have a YouTube channel linked there, and this in time and past programs are listed there. So also want to encourage you to check out the very cool stuff over here in the corner before you get out of the room. But thank you so much for coming tonight. It's been a wonderful night.